Okay guys, welcome back, and let's pick up where we left off, talking about the anatomy of the kidney. Let's go into a little bit more of a microanatomy approach now and have a look at the functional unit of the kidney, which is responsible for the production of urine, and this functional unit is called the nephron. Nephrons are going to be located mostly in the renal cortex, but you're also going to see the tubules of the nephrons extend into the medulla. So let's have a closer look at that right now. Alright, so a nephron is made up of two parts. The first part is what we call the renal corpuscle, and this is going to be made up of the Bowman's capsule and the glomerulus, and this is where the blood is actually filtered. So the glomerulus is a capillary bed, and the Bowman's capsule is a place that captures everything that gets filtered out of that capillary bed. Behind that renal corpuscle, or leading away from the Bowman's capsule, is a tubule. Now this tubule is twisted and turned, and it makes it's convoluted, and it gives it a greater surface area, and that's where we get to see stuff being moved in and out of the nephron, which is going to regulate what is formed into our urine. So here's a look at a stretched out nephron. We're going to start up here. So these two structures together are the corpuscle. We get the Bowman's capsule, which is shaped as that blue C, and the glomerulus, which is that red ball of capillaries. Now right behind that in the pink here, so ending here, starting right at the end of the Bowman's capsule, that's going to be our proximal convoluted tubule. Sometimes it's called a PCT. The next loop that we see, the next section we see, is going to be this descending and then the ascending loop of Henle. So that's going to be in the green section here. And then we start with the next section of the nephron, which is going to be the distant convoluted tubule, or DCT, and that's going to be the purple we see starting here and ending right here. So that's the microanatomy of the nephron, and later on in the unit we're going to talk about what physiologically happens in each section. So you can see the pyramid, the way that the kidney is organized. We've got a very, very efficient organization so that we can maximize the number of these nephrons that we have, so that we can have a really good efficient filtration of the blood and production of urine. So first of all, just have a quick look at the way that the blood supply is organized in the kidney. You can see that we get the big renal artery that branches off the aorta, and then we also have a big renal vein which empties back into the inferior vena cava, and then the blood flows around each one of the renal pyramids. If we take that section of renal pyramid right here and we move it over like they did on the picture here, you can see how we get all of those corpuscles, the Bowman's capsule and the glomerulus. They all line up like this inside the cortex. And if you think back to the lab yesterday, you looked at the differences between the cortex and the medulla. You always found the Bowman's capsules in the cortex, which makes that cortex tissue look different under a microscope than what we see down in the pyramid down here, which is mostly striated. Well, it looks like striated tissue, because that's where we're going to see a lot of tubules. So there's quite a bit of difference in the way that's organized, and you can actually see it with your naked eye when you look at it in dissection. Now, if we go on to even greater magnification, now we're looking at a functional unit. We're looking at a nephron. The nephrons, of course, is going to be in the purple color down here. You're going to have our corpuscle right in the middle, the glomerulus and the Bowman's capsule. Immediately after the corpuscle, this section here is going to be the convoluted tubule. Then we're going to have the descending and ascending loop of Henle. After that, it'll be the distal convoluted tubule, which will finally empty into the collecting duct right there. So amongst all of that jumble of tubules that we call the nephron, you're going to see these capillary beds. So we're going to have these peritubular capillaries. blood vessels that wrap all the way around the nephron, and that's going to help us regulate what goes into, what comes out, or sorry, what goes back into the blood, and what gets counted or excreted as our urine. So it's a really, really well organized structure inside the kidney. It's a very efficient design, and it's really fascinating to look at from a physiological point of view. So this is the order of the blood supply in the kidneys. We start off with the big old renal artery that branches off of the aorta. So right here, this is going to be our renal artery, the big red one. It's going to go into an afferent arteriole, which is going to then branch into the glomerular capillaries. So the blood vessels that we see inside the glomerulus, that's going to be these guys. Once the blood is filtered at the glomerulus, it'll leave the glomerulus through these efferent arterioles. It'll travel through those peritubular capillaries that we've seen wrapping around the nephron. From there, it'll go into smaller veins, and then finally it'll exit into the renal vein, which is the big blue blood vessel that we see leaving the kidney and hooking back into or connecting back to the inferior vena cava. Now, here's a look again at a stretched out nephron. You should be able to recognize the four different sections, I guess, of the nephron now, starting with the glomerulus, the proximal convoluted tubule, the loop of Henle, and the distal tubule. One thing that I should point out in this picture here, this section is going to be in the cortex. So when we looked at the cortex under the microscope, you could always find these corpuscles, the glomerulus. You'll also see tubules, not really organized, they're kind of convoluted. So you'll see proximal and distal tubules in the cortex, but the loop of Henle usually extends, and the collecting tubule as well. So this section here is what we're going to see in the medulla, which is why we kind of see a disorganized pattern in the cortex, but we see a fairly uniform pattern of tubules when we look and we see the medulla. So what the next, or the last part of this unit is going to be dealing with is directly this. We're going to look at the physiology, how these things here, you can see the different ions, you can see biologically important molecules like glucose, amino acids, bicarbonate, water. We're going to look at how all of these things move in and out of the proximal convoluted tubule, so how it gets into and out of our blood, and how it gets into or out of our urine. Now, individual nephrons, we start off with the corpuscle, we move into the proximal convoluted tubule, the loop of Henle, and then the distal convoluted tubule. Those are all going to attach to another tubule called a collecting duct. Now, collecting ducts are organized so that they have several nephrons emptying into them. And each collecting duct is going to empty into a papillary duct, which will empty into a minor calyx, which will enter into a major calyx, which will enter into the renal pelvis. So this is the way that the structure is organized, and I'll show you some pictures of that in a couple of seconds. And along the way, we make our urine more concentrated or more diluted depending on the state, the metabolic state, or the health of our body at that time. So here is a good picture of the way that the nephrons are organized. We can see we've got our afferent arteriole right here, the red one. The blood vessels go into the glomerulus, into the capillaries of the glomerulus. The blood is filtered. The stuff that exits the blood collects in the Bowman's capsule and then travels through the proximal convoluted tubule, down into the loop of Henle, and then back into the distal convoluted tubule, and finally into this structure here, which is our collecting duct. So if we look at this nephron here, we can see it's hooked into the collecting duct. If we look at this nephron here, we can also see it's hooked into the collecting duct. So if we have a look at the branches on the collecting duct here, we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 different nephrons can hook into that collecting duct. Now that collecting duct is going to drain into a papillary duct and then into a minor calyx. So this is how the nephron is structured. You can also see here from the branches on the afferent arteriole that we can have 1, 2, at least 3 nephrons organized around that one little tiny area. So it makes for a really, really large surface area that we can filter the blood, and it makes for a very efficient system of maintaining the constitution of our blood and excreting any wastes that we don't need at that particular time.